podcast. But that said, in case you don't know me, I'm Kason now. I'm co-hosting this seminar series with Petra, and today we have Casey. Similar names, but yeah. Difference is that Casey is an Eminother group leader now in Jülich, and the lab works on multimodal neuroimaging and computational modeling to study the maturation of cytoarchitecture, connectivity, and functional dynamics across early life, but at a like a multi-stage, multi-scale approach from genes all the way to, I think, large-scale brain dynamics. And today, I think we'll hear a bit of everything of the words I put together as introduction. So yeah, happy to um,
Great.
and then yeah.
Yeah, great. Thank you so much for the invitation. Yeah, today I wanted to focus on one aspect of our research, which is really focusing on the microarchitecture. This is something we kind of take a bit of a special interest in because I think it's a little bit underappreciated sometimes or under acknowledged in the field. So I think it's really fun to kind of spotlight and talk about how this can be useful in terms of understanding cortical organization and also incorporating into computational models or just generally into our understanding of the brain and our neuroscientific analyses. So we all know neuroanatomists have recognized for over 100 years that the cortex is very heterogeneous. So if you've ever looked in a neuroscience textbook, you probably know this picture from Brodmann, the famous map of cytoarchitectural areas. What it doesn't show is how those areas really differ. So if we look in at some 1 micron scan, so if this is Nissl stained human brain tissue, and so on the left, for example, you have V1, and on the right, frontal pole. So I think even to the untrained eye in V1, you can see some layers there. So you can see that in some cortical depths, there's much denser number of cells. Sometimes the cells are bigger or smaller as well. But on the right, even a neuroanatomist, so when we have to spend like hours trying to annotate layers, neuroanatomists even struggle to find the layers on the right as well. So across the cortex, there is really vast architectural heterogeneity. And yet, foundational theories of cortical microcircuits define this principled universal construction of feed-forward feedback processing, as if laminar organization is the same across the entire cortex. But there's been a few studies recently that have shown actually incorporating architectural heterogeneity into biophysical, generative, and disease spread models. It actually improves their fit to empirical data when you incorporate architectural heterogeneity. So we can take this to mean that regional, developmental, and disease-related differences in brain function are interconnected to the architectural heterogeneity of the cortex. So the way I think about it is that the cortical architecture really constrains brain function. And there's a lot to unpack in terms of the real nature of the relationship between microstructure and function. But there's been several challenges in our way to really uncovering this. So the first is that there has been a heavy reliance on cytoarchitectural maps, such as Brodmann's areas, but this really applies to all histology-based atlases up until quite recently, is that they've all been observer dependent. So in other words, where one person draws a border isn't necessarily where another person draws a border. And this means that they don't really fit to today's scientific standards of reproducibility. And we can't necessarily rely on these to appropriately represent architectural heterogeneity. And that's aside for the huge debates in neuroanatomy about which atlas is really correct, if it's areas, gradients, etc. The next thing is that cytoarchitectural descriptions have historically been very subjective and non-quantitative. So this is a type of table you would typically find in an article about cytoarchitectural areas, kind of defines areas, and then the neuroanatomist generally describes in like very interesting detail what the differences are. But that makes it very hard to compare across regions and individuals. We don't have the numbers that we love to use in computational neuroscience. And finally, there's this, impossibility of measuring function in postmortem specimens. So meaning any detailed, really nuanced, direct investigations of microstructure function coupling have basically been impossible if we rely on histology for gauging cytoarchitecture. So this altogether has motivated a line of research by me and my colleagues and my group to look into more quantitative and data-driven mapping of cortical architecture in a way that's also translatable to in vivo MRI, so we can compare it to function and so we can look at development. So with that in mind, today I'm basically going to highlight a few studies we've undertaken to do so. So to start with, I'll talk about how we've been mapping how cytoarchitecture varies across the cortex using histology. And then I'll move on to our tests of whether we can reliably measure microarchitecture with MRI. And finally, how we can use this to examine the implications of microarchitectural differences on function and development. Okay, so starting off with histology. So I'll start by introducing you all to Big Brain. This is a 3D reconstruction of a sliced and stained human brain. The brain was extracted, sliced coronally at 20 micron thickness, stained for cell bodies, and then the slices were scanned, basically like in a usual type of scanner, and then reconstructed into 3D. And the way we can use this in 3D is kind of similar to an MRI, honestly, because the 3D reconstruction is relatively accurate and smooth. We wanted to approach mapping this in as objective a way as possible. So we devised a way, kind of inspired by previous methods in histology as well, to extract depth-wise changes in cell body staining. But important for us, it was to do this in 3D, whereas previously in histology, this had been done mostly in 2D. So this procedure basically involves first taking the outer surface of the cortex, so the peel layer, as well as the matter boundaries. So that's the purple and the yellow lines there. Then we construct a number of surfaces between these, and we just sample the cell body staining intensity as we go in a straight line down from the peel layer to the gray wet matter boundary. And this produces this nice type of profile, which shows how staining intensity relates to cortical depth. Straight away, you can see a couple of features here. You see that big bump in the middle on the far right. That is layer 4, where you have really dense granule cells. And also you see this very fast increase in the highest cortical depths. So that's the transition from layer 1 to, you don't have many neurons at all in layer 1. So that's why there's no staining there. And you very quickly go to layer 2, where you do have some neurons. So to this, it was very similar to what we've seen in 2D, but it shows that we could do the same approach in 3D. And we could apply this across the entire cortex. We had like 320,000 vertices, so really dense sampling of the entire cortex. And then we parcellated this and correlated each of these profiles to see how different parts of the brain how the shape of these profiles are similar to each other. And in effect, you have something that's a correlation matrix. And just as a few examples, some like seed-based correlations here. So you see V1, for example, its cytoarchitecture is quite similar to other sensory motor areas that are highlighted in red. And you see the same for S1, primary sensory area, also similar to the extended primary sensory area, as well as somatosensory and visual areas. Whereas on the right, you see anterior cingulate, for example, is quite similar to other areas of the cingulate and the insula. Whereas on the bottom right, you see the temporal pole is more similar to kind of a general areas across the lateral temporal lobe as well, up into the temporoparietal junction. So depending on where you see that there's different kind of patterns of cytoarchitectural similarity across the cortex. So to better understand the general pattern of this, we perform diffusion map embedding. You can think of this as kind of like a nonlinear principal component analysis. And we were first interested in the first component, and that's what you see here. So this is the first eigenvector. So more similar colors mean that you're more similar in terms of your cytoarchitecture. And it basically goes from these bluey colors in sensory areas and kind of motor areas, primary motor areas as well, through association areas up until red, which are these more limbic areas. And this to us immediately conjured memories of classic neuroanatomical texts, for example, by Sinides or Meslem, who talked about a sensory-fugal axis. So that you see this stepwise change in laminar organization going from the very structured, clear layers of sensory areas to these more agranular areas in limbic areas. So using this data-driven approach, we could basically recapitulate what neuroanatomists had been observing for decades. But what's been really exciting lately is that we more recently focused in on association cortex because it was already well established. Yes, sensory areas are very different to limbic areas. But what we didn't know was how this really maps out in functional networks that are, more novel conceptualization and more new conceptualizations as well. So we focused in on the default mode network, which I assume most of you are familiar with in this seminar. And what we found that was really interesting here is that, so not only are there, I should explain this figure very quickly, actually. Here, once again, we took the first principal component, the first eigenvector, from a dimensionality reduction on the cytoarchitectural profile. So it's using the same data as the one on the left, but just focused into the default mode. And now you have blue and red as the color map of that first eigenvector. So what we see is essentially there's two kind of types of cytoarchitecture within the default mode. Let's say the blue type and the red type. Now, what you'll notice is that all the subregions of the default mode have both blue and red subtypes within them. So when we first started this project, we thought there was a high chance that you'd have certain subregions that are one type and different subregions that are another. But what we found instead is that what makes the subregions distinct is sometimes the proportions of each cytoarchitectural type they have, but also the patterns within them. So for example, the parahippocampal area of the default mode has this very smooth gradient from red to blue, whereas the prefrontal cortex has this interdigitated pattern. It really fluctuates very quickly between these different types of cytoarchitecture. And we're hypothesizing the moment we want to follow up on this as well, is that this is related to the different forms of information processing in these areas, because the neighborhood that you're in the brain is very important for the type of information that you're receiving. Like the vast majority of connections in the brain are just nearest neighbor connections within the cortex, I should say. And so a gradient means you can have a gradual convergence of information from one type of processing to another. But in the prefrontal cortex, where you have this fluctuating pattern, that is related to perhaps these studies that we've seen of the interdigitated pattern of contralateral versus ipsilateral connectivity of the prefrontal cortex, where you're receiving information from different types of association zones in the brain. So for example, you could be receiving contralateral prefrontal information, and then in the next little area over, ipsilateral parietal information. And by putting those directly next to each other in the prefrontal cortex, you can combine very distinct types of information. And that contrasts with this gradual convergence that we see in the parahippocampus. So there's different patterns of cytoarchitecture across the default mode could be related to different types of information integration that's occurring across the subregions of the default mode. And I think speaks to the possibility that that's what makes it useful for the default mode to contain all these different areas, because they're contributing something different to the overall integration of information. If you're interested in using this histological data, we wrapped a lot of it up into Big Brain Warp. It's also a toolbox for doing transformations between histological and vivo imaging data. But we've also released like the profiles and the gradients from there. So if you're interested in this, feel free to go to the GitHub or the read the docs or email me as well. Okay, the next section I want to talk about is how we actually translated this approach to in vivo imaging. So this 3D approach was actually very straightforward to apply to MRI, but just because you can do it, does that really mean that we should do it? So we knew that depending on the type of MRI you apply this to, it would reflect different types of biological sensitivities. So if you have an R1, we know that's highly sensitive to myelin. If you apply it to an R2 star, for example, that would be sensitive to iron. So it should provide some insights into the cellular makeup of the cortex, but it's really specific based on the type of imaging modality that you'll be using. And you'll also notice there that the profiles are going to be much smoother because of the resolution, but I'll get more into that as I go through it. We recently released this entire procedure as a toolbox called CortPro. It's up on bioRxiv at the moment, and it's really just one line of code to run everything. So you can basically direct it to the type of data you have. The most basic form of this is just point out here's a T1 and here's a T2, and it will create the T1, T2 ratio for you and construct the PL on white matter surfaces using FastSurfer. But you can also skip these steps. You just really indicate it to it where the data is, and it sorts out the rest. So it's meant to be as straightforward as possible, but essentially it runs these steps of compiling a microstructure sensitive image. So it's something we're going to sample the intensities directly from, reconstructing the PL on white matter surfaces, and then registering these to the same space. Additionally, it's going to generate equivolumetric surfaces between the PL on white matter surface. The important thing of this being equivolumetric, maybe this is a little bit technical for some people, but I think it's quite cool, is that the layers actually kind of bunch together depending on if you're in a gyrus or a sulcus. So you can think of this, I like to think of it as like a martini glass. So the centimeter on top of a martini glass carries so much more liquid than the centimeter at the bottom. And so to compensate for this in a way, the way that we actually see cortical layers is that they spread out depending if they're in a gyrus or a sulcus to maintain the volume rather than the depth. And so in past work by Varnot et al., they've shown that using this approach where you modulate the depth based on the curvature, you can approximate this equivolumetric model and you actually stick to the cortical lamina much better than just using equidistant model. So that means that if you're in a gyrus and you're looking at a sulcus at the same cortical depth in our profile, it means they're corresponding to a similar cortical layer because we've tried to accommodate for these oddities of curvature. The other big question that often comes up here is about the resolution of the data and whether we can, how many voxels are we really sampling from here? So if you do a quick calculation on a standard like quantitative T1, for example, of 0.8 millimeter like resolution, isotropic, we have average cortical thickness of like 2.8, then you'd guess you just have like 3.2 voxels within the cortex. But we know that, you know, voxels aren't perfectly aligned with the cortex as well. So we ran a bunch of tests on some data that we had of how many voxels really are within each profile. How much information can we really sample here with standard like research quality MRI? So on the left, you see if you just do a nearest neighbor approach, so you really just sample the exact voxel that you land on with each surface, then you actually have much more than that 3.2 on average. It's closer to 8, but it reaches all the way up to 13 or 14 in thicker parts of the cortex as well. And so this is to do with the orientation of the brain in the voxel field. We typically apply a trilinear interpolation though, so that means that you take a weighted average of the whole neighborhood of voxels. So it's more like taking a column rather than a single line. And there we see a huge increase in the number of unique values that you get as you move from peel to white matter boundary, more up to like 40 unique values within that profile. So typically we kind of strike a bit of a middle ground in how densely we sample this. We've adopted like 14 as a typical standard. That seems to be like the most you would get out of very thick cortex, but it is somewhat oversampling other areas of the cortex. That's something to keep in mind. But generally, with the analyses that we've done, it is quite robust to changes in the precise number of surfaces that you use, but this is a parameter that you can just change in CortPro as well. The way that we often analyze this is using central moments because the profiles themselves, as I said, the sampling, there is some redundancy there, obviously. And so looking at, region-wise differences, for example, between every cortical depth is just going to be, a lot of redundancy there in your model. So instead, we look at the shape of the profile, typically. So the 0th first and the second central moment are shown here. You see their regional variation on the left, and then using a sliding window approach, what a profile looks like with a low to high 0th moment, for example, or first moment. And then on the right, you see a correlation between the profile property and the regional variation. This basically gives you an overview of what does it mean to have a higher or low 0th moment. So the 0th one is it's unrelated to the shape of the profile. It's just the mean shift. It's actually just the average of the cortical profile. The first moment is the center of gravity, so it's the balance between the upper and lower layers. And the second is the concavity. So it tends to be whether you have a flat profile or a bendy one. So what's been interesting here is though, typically you see that, sensory regions are quite distinct, but the way that these different moments vary across association cortex is very different. So they really provide complementary insights into profile shape. And as you move to higher moments, though, they start to become collinear. So we've found that at least for standard in vivo imaging data, these are the three that provide unique and complementary information. So we can, clearly extract these profiles, we can analyze them in ways, but we wanted to test, like, is this really sensitive to a lamina itself, actually? Like, what's the validity of this? So we tested with this really amazing data set where they scanned one individual 53 minutes for each scan. And it was performed eight scans, 53 minutes each. Apparently, he is really good at not moving in the scanner as well. And so together they upsampled this image to 250 microns. So for an in vivo MRI, that's just pretty incredible. But what we could do with this is construct the profiles and then downsample the image and redo the procedure again. So you can see in the dark blue there is the really high resolution original data. You see really nice bumps in the profile relating to different aspects of, like, essentially like the one on the right, the DLPFC. The big bump there in the middle is probably just below layer 4, for example, where you have like higher myelin kicking in as well. Or sorry, that's the bump there. And essentially, as you downsample the image all the way to like 1 millimeter, you still have some aspects of those bumps conserved within the shape of the profile. And if you look at the moments, so the 0th moment doesn't change at all. That's ambivalent to shape. But the first and the second moments, if you look at them across the whole cortex, they're really highly correlated, even as you downsample to 1 millimeter. It's still, sorry, the font is pretty small there, but it's still a correlation of 0.95 or 0.96 for the first moment and 0.9 for the second moment. So even as you move down in resolution, you're still sensitive to differences in laminar organization across regions of the brain. Next, we looked at the reliability of this approach. To do so, we used a data set where 17 people were scanned 2 times within one day. They were removed from the scanner and then placed back in for each session. And they did this for both MP2 range sequence producing an R1, so a quantitative T1 image, as well as for T1 weighted over T2 and T2 weighted. So we could look at these two very common types of approaches for looking at myelin essentially across the cortex. So what we see is there's extremely high reliability across the sessions, with the exception of the 0th moment of T1 over T2. So you see at the top, it's just the intersession similarity, and on the bottom, the intraclass correlation coefficient. And the intraclass correlation coefficient is per region, so you see it's pretty consistent across the brain, with the exception of these real big jump down for the 0th moment of T1, T2. So this isn't so surprising, really, because we know that T1, T2 is affected by the inhomogeneity of the transmit field. So you can see an image of that in the greeny brains there. It's even stronger at 7T. So this means that the intensities that we're sampling are biased based on where they sit in your field of view, essentially. And these transmit field biases are influenced by the specific scanner that you're using, body size, and also positioning. Positioning is an interesting one, I think, because, for example, there's one subject there who the intersession similarity is really low. You might see it's like down near 0. My suspicion is that this person just got back into the scanner in a very different way with their head a bit tilted. And then you see that the T1 over T2 values, like the transmit field bias, is completely shifted on them. There was also like quite a nice paper by Matt Glasser, who first kind of developed this T1, T2 ratio as well, saying that this is like a known problem and there are potentially ways to create like kind of pseudo fields to remove this effect and everything as well. But I think that's still an active area of research. And generally, my suggestion is always to collect an MP2 range instead, because it is already corrected for these types of problems. And that means you can use the mean and compare it across individuals as well. And we found the same result in 2 other data sets with MP2 range sequences as well. And then taking this a step further, we looked if the profiles are robust to differences across studies. So for example, we looked at ABCD, where they had 5,000 subjects made it past our quality control, taken from 21 different sites across the US. They're all roughly the same age because we use the baseline of this data set. So all the people in this study were around the age of 10. And across the 21 sites, they also use 3 different scanners. So it's like Philips, Siemens, and GE, and also different subtypes of each of those scanners. So huge discrepancy across the sites. And yet we see that, and this is using T1 over T2, I should say. We still see that the first and second moments are really similar if you look at the group average cortical maps across all those sites. You see a drop in the replicability for the 0th moment, though, which once again is likely related to these non-biological or noise-related effects of the transmit field bias on the T1 over T2. And finally, we also saw that even there's some pretty good replicability across different modalities. So in the shams dataset, because we had MP2 range as well as T1 over T2, we could compare within each subject and within each session how similar are the maps that you produce from these profiles. Yeah, I'm sounding like a broken record now. It worked really well for the first and second moment, not so well for the 0th one, because yeah, once again, transmit field biases, but I think this is a good take-home message because it means that you can actually use the T1 over T2 across different studies. As long as you're looking at these local ratios, essentially. So the reason that the first and second moment still are replicable and reliable, even with T1 and T2 over T2, is that you're looking at differences, the relative differences within a very small patch of the field of view. And the transmit field biases are actually very low frequencies. They change very, very little within one patch of cortex. That's why you can look at the shape of the profile and not be affected by these biases. Yeah, so with that, I hope I've somewhat convinced you that this is a very reliable and valid measure to use. And I'll quickly touch on some of the interesting ways that we've used this to try to link microstructure with function and start to investigate developmental changes. So to start off with, in one study, we wanted to understand whether microstructural similarity, as well as other structural metrics, how they contribute to differences in functional connectivity across the cortex. So using these profiles as well as geodesic distance and diffusion-based tractography, it tried to explain variance in functional connectivity within a machine learning framework. What we found is that each of these structural metrics were very similar in terms of their the model accuracy. So explaining about 50% of variance in functional connectivity in the test set. But we saw a big jump up in the accuracy of the model when we included all of them together. So the take home here would really be that microstructural similarity is adding information in terms of the functional organization of the brain beyond distance and beyond the structural connectivity as well. And that just emphasizes its value in for understanding the functional organization of the brain, I think. We also are interested in understanding at an individual level how microstructure and functional gradients are interrelated, because there was a lot of discussion with the functional gradient work that this was recapitulating Meslam's cortical hierarchy. But when we saw the microstructural gradient, we thought that actually aligned better. So we wanted to compare them directly, and we could do so at a single subject level using this approach. And so we found that microstructural and functional gradients were basically moderately correlated across subjects, but there was some individual variation there. So the average correlation was about 0.5. What I find more interesting, though, is actually the divergences here. So if we look at how functional networks are placed across these gradients, so the colors are shown on the right if you're not familiar with the Euro colors scheme off the top of your head. But basically the somatomotor and visual networks are pinned at the bottom of both of these gradients. So as I've said, sensory motor areas are very distinct in terms of their architecture and their function. We all know that. But what was quite interesting is that while the default mode and the frontoparietal network occupy the apex of the functional gradient, they're actually distributed quite broadly along the microstructural gradient. So in other words, that means that they encompass a broad range of architectural heterogeneity. So you see, yeah, this is especially true as you see here in this spider plot for the default mode and frontoparietal network, which are notably like highly expanded in humans. I find this really interesting because often we think of functional networks as quite homogeneous in a way, like they're very unified networks. But what this work started to show is that part of being a functional network is capturing a broad range of cytoarchitecture within them. And I think this is related to minimizing redundancy in the brain as well. Like, what's the point of a functional network if all the components are the same? So I think that's something we kind of followed up in actually with this paper on the default mode network, where we once again were able to translate these more fine-grained patterning to in vivo MRI. This was more of a challenge because we were needing much higher resolution data to look at distinctions within the association cortex. So we had to use 2 sessions of 70 MRI per person at a 0.5 millimeter resolution. But in doing so, we could essentially replicate what we found in histology, where you have different types of subregional patterning within the default mode, either this gradient within parahippocampus, for example, or interdigitation within the prefrontal cortex. And we specifically tested this using individual specific default mode networks as well, rather than just applying an atlas. And just the final couple of slides, I want to gear shift a little bit into neurodevelopment because I just find it a super interesting area of research as well. So we're trying to understand how does microarchitectural heterogeneity really emerge? And so in one study that we did a few years back, actually with colleagues at Cambridge, with Richard Bethlehem and Ed Bulmore was a senior author on this paper, actually. Oh, he's not at Cambridge anymore, but he was then. So we were looking at age-related changes in the microstructural gradient in late adolescence and early adulthood. And we found that generally you see this principal sensory-fugal axis already quite stable by the age of 14 years across all the subjects. But there was a change specifically in the middle of this gradient. So the sensory areas were already very distinct, the limbic areas were already quite distinct, but this expansion was happening in the center that you had parts of the brain. So, for example, the red in the top row, these prefrontal areas, which were becoming in a way more sensory-like because of the change specifically in deeper layers of the cortex in terms of how they were myelinating. And then you had other areas of association cortex, like you see in more these temporal areas, they were becoming more limbic-like because they were myelinating in a different way. And so it seems that while those anchors were already there by early adolescence, throughout late adolescence and early adulthood, the association cortices are, as we know, still developing, but not in one direction. I think was really interesting to see here is that they might be bifurcating in a way, developing different roles and becoming more varied in themselves as well. And the last study I want to highlight was just something from my PhD students. It's up on bioRxiv at the moment where we looked at baby brains in the DHCP. This was a challenge as well because obviously the baby cortex is very thin, but we looked at whether we could still look at changes in the cortical profiles based on both gestational age, because as you see in this graph on the left, there's a wide range of gestational ages at which the babies were born. So ranging from 24 weeks all the way up to full term, kind of 41 week old infants. And also how long after birth they were scanned. There was a range of about 1 to 7 weeks after birth that they were scanned. And we started to weigh up what are the contributions of changes in the cortical profiles based on whether you were in utero for longer or if you're ex utero for longer. Because you obviously have these very dramatic shifts in this more innate kind of programming of cortical maturation that's happening in utero to huge sensory stimuli that's occurring ex utero as well. The lengths are a little bit different, but from what we saw, so you see that the gestational age had a really large effect regardless of postnatal age. So in the top column, in the top rows, you see the changes in the first and second moments, really varied patterns. I won't get into it too much here but there was different changes in the profiles across the cortex that withstood correction for the postnatal age. So there's really strong effects of a length of like the duration in utero. Whereas the effects of postnatal change were smaller, it seemed. And maybe that's a matter of just not capturing enough of that postnatal period as well. But there's obviously a lot of cortical maturation occurring in that last trimester as of pregnancy as well. So this has been really exciting to start seeing come together. Okay, so just to wrap up, some of what I've learned from this is that Based on the histological studies we've seen, there's really fine-grained differentiation of cortical architecture, which is embedded within large-scale gradients. And we've seen that this really challenges simple area-based modularity. Maybe makes us think about different ways to characterize nodes in computational models, for example, rather than just saying it is a distinct parcel or it has a certain level of myelin. I think we should be moving towards more detailed, perhaps, descriptions of what makes a parcel unique in terms of not just its the the layer thickness or something, but also the makeup of the pattern of cytoarchitecture within that area, for example. In terms of method, we've seen that it's really a strong validity, reliability, and replicability of this approach with in vivo MRI, especially if you're using quantitative sequences. There's a little bit more caution regarding just using T1, T2 if you're focusing on the mean, but you can definitely use the profile shape from T1, T2 based on these experiments. And finally, we definitely see the microarchitecture has a unique contribution of functional organization of the brain. It's being continuously sculpted throughout maturation, but generally there's so much left to explore in these domains, and I think we now have the foundation to do so. So with that, I just want to acknowledge many of the people involved in this project, especially at the moment, I'm based in Einem 7 at Forschungszentrum Jülich, which is the pretty picture there. A lot of this work was done during my postdoc over at the MNI with my mentor, Boris Bernhardt, as well as all my collaborators, especially Johnny Smallwood, who was incredibly instrumental throughout this work.